Hello, everyone. Welcome to our next presentation titled Synthetic Biology Approaches to Improve Cell Therapy Function. I'm Amanda Grimm, your GenScript moderator for this session. I'm thrilled to introduce our speaker, Dr. Michelle Hung from GeneFab. Dr. Hung has over 12 years experience in mammalian synthetic biology and engineering next-gen cell and protein-based cancer therapeutics. In her current role, she leads GeneFab's research collaboration with Senti Bio and Rock Therapeutics, building next-gen cell therapies for regenerative medicine. In her presentation today, she'll provide you with details on how her team can support the development of safe and efficacious gene and cell therapies, as well as showcase their innovative solutions to design custom CARs for NK therapy. Remember, you can submit questions at any time during the presentation using the Q&A box. So let's go ahead and dive into our main presentation. Dr. Hung, please take it away.
Hi, everyone. My name is Michelle Hung. I'm a principal scientist at GeneFab, and I want to thank the organizers at GenScript for giving me the opportunity to talk to you today about synthetic biology approaches to improve cell therapy function. So there has been really an explosion in the number of innovative cell, gene, and nucleic acid therapies over the past years. And these types of medicines are many times more complex than small molecules or biologics, which means that there are vastly different challenges for the design and manufacturing of these types of genetic and cell-based medicines. This includes viral particles, nanoparticles, human cells. They just contain so much more in them than some of our more established forms of medicine, like small molecules and proteins and antibodies. And so there really is a need to support these really complex medicines. And that's where GeneFab comes into play. So GeneFab was created to overcome limitations within the current CRDMO ecosystem. For example, drug candidate selection. Currently, the genetic design space is nearly infinite, and homegrown approaches are inefficient and slow. GeneFab has platforms for high-throughput synthetic biology, bioinformatics, and AI, and design services provided through our expert R&D team, which I'm going to talk to you about today. Additionally, when you need first-in-human data, there's a high risk when R&D programs are just thrown over the wall to manufacturing, and there's inadequate CDMO support for early-stage programs. And GeneFab was built to kind of overcome this by really partnering with clients and creating an end-to-end cell and gene therapy GMP platform. And when you get to commercial scale, there's really heavy capital investment requirements, high cost of manufacturing. And GeneFab is a multi-site network with an internal R&D focus on cost reduction to help with that as well. Today, I'm going to talk to you about synthetic biology services, and we have synthetic biology solutions to a lot of key challenges in cell therapy. So one key challenge in cell therapy is precise disease targeting, making sure you are hitting your on-target cells and not your off-target cells. And to support that, we can perform high-throughput screening of CAR binders and different architectures of CAR to help customize a chimeric antigen receptor that's exactly fit for your purpose. We can also help humanize CAR binders that are based on rodent scFvs. And we can also develop cell type and cell state selective promoters and UTRs to enable you to express things only in one specific cell type or cell state. When it comes to maintaining the cell phenotype that you desire, as well as the therapeutic function, our team is highly experienced in protein engineering, which can help to control immune cell phenotype and cytokine secretion by expressing exactly the right protein that you want under the right conditions. We can develop synthetic master regulators of cell state, and we can customize our immune cell functional assays to really look at exactly what you want to evaluate. And to enable you to access therapeutic windows, we also can do high-throughput screening of protein and small molecule binding domains. We can control protein stability, transcription, and apoptosis to allow you to express exactly how much payload you want at the time that you want it. GeneFab's synthetic biology team is a problem-solving task force with a diverse set of expertise and committed to solving your challenges. So the way that we generally work with clients is clients come with a vision and a current technology, potentially some data and methods. And the Synthetic Biology Task Force at GeneFab comes with some initial parts and know-how. We onboard your project. We analyze and or generate input data. This could include bioinformatic data on transcription factor profiles, for example, or gene expression in your cell types of interest. We'll establish the cell culture exactly as best to model your system. Then we'll design libraries and rational designs to achieve your goal and design custom functional assays to determine whether different parts are actually achieving your goal. We'll perform high throughput screening, and we'll optimize and validate using machine learning to help inform the design of even better candidates. And this will ultimately lead to your next gen therapeutic. And to demonstrate our success in doing projects like this, we'll talk about GeneFab's previous ability to deliver on various collaborative projects. So GeneFab's synthetic biology team has delivered on diverse projects for leading cell and gene therapy companies. And a lot of this work was done when the scientists at GeneFab were a part of Senti Biosciences. We've now split from Senti Biosciences and are the synthetic biology team at GeneFab. We've supported Senti Bio in custom gene circuits for boosting CAR-NK safety and efficacy. We've supported Rock Therapeutics for control of cell behavior for regenerative medicine, including autonomous cell state control and small molecule regulated cell death. And we've supported Spark Therapeutics for custom promoter discovery for AAV gene therapy. So in our support of Senti Bio had kind of the initial platform of a CAR-NK cell therapy. They had an engineered NK cell platform. They had tumor antigens of interest. They had some binders from rodent immunizations, and they had an inhibitory NOT gate CAR architecture. And the idea that they ultimately wanted to achieve was to have a logic-gated CAR-NK therapy, where their CAR-NK cells can kill multiple different kinds of AML types of cells, including blasts and LSCs, which have different antigen expression, CD33 and FLT3, while preserving healthy hematopoietic stem cells, which also express CD33 and FLT3, but potentially also a safety antigen. And they needed synthetic biology support in terms of optimizing CAR expression, because they wanted to be able to target two on-target antigens as well as their NOT gate that would protect cells expressing a healthy safety antigen. They also needed the design of the OR-gated architecture, so a CAR that could target two antigens at once. They needed a selection of the safety antigen that would be expressed on healthy HSCs but not on the AML cells. And finally, selection of which binders would be most optimal and humanization of those binders. And so the team of scientists who are now at GeneFab took on this project to optimize CAR expression, develop the OR-gate architecture, discover safety antigens, and humanize binders. The design process involved many factors, including looking at different tumor expression profiles and using bioinformatics to identify ideal safety antigens, as well as looking at existing validated binders and altering which binders to try, which order to express them in, what linkers to use, what intracellular domains to use for the CAR, as well as how to humanize them. And all of these different variables were combined and screened in a high throughput fashion, ultimately resulting in the design of therapeutic CAR-NK cells that express high levels of a humanized OR CAR that's able to kill cells that express either CD33 or FLT3, and a NOT CAR that's able to protect hematopoietic stem cells. And this ultimately expands the number of patients that Senti can treat who can benefit from this AML cell therapy. Another story from our work with Senti Biosciences is the engineering of a master regulator. So again, Senti had this CAR-NK platform. They had their engineered CAR-NK cells. They had validated OR and NOT gates, but they needed to enhance NK cell persistence in order to maintain that therapeutic effect. And so they wanted a master regulator. And the idea that we ultimately designed is called calibrated release IL-15. This idea came around from the need to synthetically enhance the NK cell survival and killing, and IL-15 is really known to help with that. But we wanted to both provide secreted support for nearby NK cells and also membrane support or autocrine support for the NK cell itself that's being engineered. And so the idea was to design an NK cell master regulator, which involved a certain amount of IL-15 being expressed on the cell surface, but at some rate, this IL-15 is cleaved off the cell surface by a membrane-based protease and released into the supernatant to provide support for other NK cells nearby. So they needed a design that would achieve this balance between membrane-associated IL-15 expression as well as secreted IL-15 expression, would be compact and highly expressed along with the other CAR genes in the vector. And so the team of scientists who are now at GeneFab designed these synthetic calibrated release IL-15 constructs, these proteomics informed design to design a suite of different potential protease cleavage sites, and then a literature informed design of many different transmembrane domains, and combine these into a small library, perform high throughput screens to identify the best construct that gave high membrane associated and high secreted IL-15 from a single compact transgene, enhanced NK cell persistence in vivo, and also enhanced performance in serial killing assays, ultimately resulting in a custom master regulator that can fit in the existing Senti or not CAR vectors and greatly enhance CAR NK persistence and killing in vivo and in vitro. Additionally, another example of the GeneFab services involve the development of a regulator dial. So again, in support of Senti Biosciences, the initial technology that Senti Biosciences had was this regulator dial that could control protein expression based on the presence or absence of a small molecule. So in this system, you'll see on the left-hand side how the system works. In the absence of a small molecule drug, there's a DNA binding domain linked to a transcriptional activation domain, and there's a protease that cleaves the linkage between the DNA binding domain and the transcriptional activation domain, such that when the DNA binding domain binds to its promoter, it is unable to turn on expression. On the right-hand side, in the presence of a small molecule drug, an NS3 inhibitor in this case, which is a FDA-approved small molecule, it inhibits the protease activity such that when the DNA binding domain binds to its promoter, it also brings along that transcriptional activator, and now you can express your protein of interest, in this case, a cytokine payload, and turn it on. And so the idea is that you could then create a therapeutic window for really potent cytokine payloads such as IL-12 to enhance the activity of a CAR-NK therapy. However, on the rightmost side, you can see the activity of this initial regulator dial, where the drug used to turn on the regulator dial was acinaprovir, which is an NS3 inhibitor. And the maximum concentration in humans is shown on the dotted line. And then the activity of the transcriptional switch is shown on the y-axis, where increasing amounts of fluorescent reporters show that the switch is turning on. And you can see that the switch only starts to turn on at concentrations well above the expected concentration in humans. So this wouldn't be a therapeutically relevant switch. And so there was a need for the synthetic biology team to engineer this regulator dial to make it therapeutically relevant. And so here the team designed many different variants of the regulator dial, varying the transcription factor activation domain, the transcription factor DNA binding domain, the linkers, as well as the promoters they bound to, and also screened a number of different NS3 inhibitor drugs. And through many screens, you can see we found a regulator dial that performs as shown on the graph in the middle, where the x-axis shows the concentration of grazoprevir, another NS3 inhibitor drug, and the dotted lines indicate the minimum and maximum concentrations expected in human serum when treated with this drug. And the y-axis shows IL-12 production from NK cells at different concentrations of drug, and you can see that there's high production of IL-12 in between the therapeutically relevant concentrations that are expected in human serum. So ultimately, the synthetic biology team was able to develop a clinically relevant switch that could control IL-12 production in CAR-T and CAR-NK cells, regulated by an FDA-approved small molecule. So now I'll move on to show how the GeneFab synthetic biology team has supported Rock Therapeutics. So Rock Therapeutics has the technology of a cell plus gene platform that includes highly specific cells manufactured from pluripotent stem cells under rigorous process control, and genetic engineering to enhance the cells with specific functions to improve their therapeutic effect. Their long-term vision was control of cell state and payload expression. And this composed 3 different projects, including safety switches, where a small molecule could regulate cell death, such that if the cell therapy was malfunctioning or causing some kind of toxicity, it could be turned off through administration of an FDA-approved small molecule drug. An additional component of their vision was master regulators. They wanted to be able to synthetically lock a macrophage-based cell therapy into a desired polarization state to combat native macrophage plasticity of switching between inflammatory or anti-inflammatory phenotypes. And finally, they wanted cell state promoters that could drive gene expression only in either inflammatory M1 macrophages or anti-inflammatory M2 macrophages. And so these synthetic biology needs were addressed by the GeneFab team, and I'll show you in the following slides. So first, I'll talk about synthetic biology support to develop safety switches to control cell therapy. So here the vision was a small molecule activated switch that could kill greater than 95% of cells within less than 24 hours. The initial technology involved taking parts from the literature, including an inactive monomer of caspase-9 linked to a tamoxifen metabolite binding domain called ERT2. In the presence of tamoxifen metabolites, the ERT2 domains dimerize, bringing together the 2 caspase-9 monomers, which form an active dimer that kick off the apoptotic cascade. We also developed a custom assay that involved using IncuCyte to evaluate the activity of this safety switch over time. So initially in the literature, people use flow cytometry to evaluate cell death, but this is really noisy and provided only a single time point. So we optimized to evaluate over time the switch activity and provide precise time course data. From this, we found that at FDA-approved concentrations of tamoxifen, killing is very inefficient, with less than half the cells dying over the course of 72 hours. So we collaborated with a company called Applmex to do computational modeling of the ERT2 binding domain and to identify computationally potential mutations to that binding domain that could increase its affinity for tamoxifen metabolites, while not increasing its affinity for estradiol, which it currently doesn't bind, and we don't want to have any off-target effects during human menstrual cycles. Based on these computational predictions, we screened in vitro hundreds of mutations and chose mutations that improved the sensitivity of the switch to tamoxifen metabolites. Then those hits were combined into a combinatorial library and screened. Here we used pooled screening followed by PacBio sequencing to identify the hits that led to the most improved sensitivity to drug. So here we're showing the results of the most optimized kill switch developed from that screen, where the ERT2 mutant that performed the best is shown here. And on the x-axis, you see time after addition of the tamoxifen metabolite. The y-axis shows killing efficiency, and that dotted line is the maximum detectable killing in the assay. And at an FDA-approved concentration of tamoxifen, you can see complete killing is achieved by our most optimized ERT2-based switch in 18 hours, whereas the original ERT2 never achieves complete killing. And so the team at GeneFab was able to develop a safety switch enabling rapid control of cell therapy viability at physiologically relevant concentrations of tamoxifen metabolites seen in patients taking the FDA-approved doses of tamoxifen. The next project that we undertook for Rock Therapeutics was the design of master regulators that could synthetically lock macrophages in an inflammatory state, combating the macrophage's native plasticity of switching between inflammatory and anti-inflammatory state. To design this master regulator, we screened a library of master regulators designed to include transcription factors, microRNAs, engineered cytokines, and receptors. And we also developed a custom protocol for transducing and phenotyping macrophages. This involved custom macrophage transduction protocol, a custom polarization time course, and custom phenotyping assays. Additionally, as a result of these screens, we were able to develop a custom screen where we look at the polarization over time of macrophages that either are engineered with our master regulator or are not engineered with our master regulator. And then we have a custom phenotyping assay that includes 11 different biomarkers to assess macrophage polarization state. So on the left, you can see our screening assay. And on the top is an example of unengineered macrophages. They start in state 1, which is M0, not polarized. In the presence of interferon gamma, they polarize to an inflammatory M1 state. And over time, if you continue to provide that interferon gamma Q, they stay in that inflammatory state, as indicated by state 2. However, if you remove the interferon gamma Q, they become less inflammatory, as indicated by state 3. And on the right, you can see these states mapped out on a principal component analysis that incorporates these 11 different biomarkers, which include cytokines and cell surface markers that tell us about the cell state. So in the gray circle, you can see the initial M0 not polarized cells, state 1. State 2 is in that red circle, which is the target inflammatory macrophage M1 state, and state 3 has moved outside of that red circle and is less inflammatory. On the lower half of the custom screening assay, you can see engineered master regulators can lock the cells in the inflammatory state. So again, the cells start out in this M0 state, but now they've been engineered with a custom master regulator. You can see that this M0 state, state 4, is very close to the state 1, so our master regulator is not affecting that M0 unpolarized state. However, upon the addition of interferon gamma, if you maintain the interferon gamma Q, the cells will be inflammatory as expected in the target state. This is state 5, which is again in that M1 circle. However, if you remove the interferon gamma Q, the cells now remain very inflammatory, as indicated by state 6, which is still in that target M1 circle, as opposed to state 3, which are cells that don't have the master regulator and become less inflammatory upon the removal of interferon gamma. And so we were able to develop a custom master regulator that maintains the cells in an inflammatory state, even after the removal of inflammatory cues. BlueRock was also very interested in being able to express payloads specifically in inflammatory M1 macrophages or anti-inflammatory M2 macrophages. And they wanted promoters that would drive the expression of their payloads only in those cell states. So that we took the approach of looking at differential transcription factor profiles and developing a library of tens of thousands of different transcription factor binding site arrays and linking those to minimal promoters to drive expression. We also looked at attack-seek based data and identified putative enhancers, which are regions of open chromatin that are more accessible in the cell state of interest as opposed to off-target states, and developed a library of native enhancers linked again to minimal promoters, which could potentially drive cell state-specific expression. And here I'm showing the outcome of our screening, where the x-axis is the strength of our promoters normalized to the EF1 alpha promoter, where the dotted vertical line at 100 shows 100% strength of EF1 alpha, and the black dot there shows the EF1 alpha constitutive promoter. The y-axis shows the selectivity for the on-target state versus the off-target state. The red dots are M1 promoters, the blue dots are M2 promoters. And so you can see that through massively parallel screens, as well as iterative designs and engineering, we were able to identify promoters for both states that have over 100-fold state-specific expression, so 100 times stronger expression in the target state than in off-target states, and strength greater than the EF1 alpha promoter, enabling strong and specific expression of therapeutic payloads in M1 or M2 macrophages. Finally, I'll tell you about our work with Spark Therapeutics on the discovery of cell type-specific promoters to enhance gene therapy safety. Spark's technology platform is an AAV gene therapy platform, cutting-edge AAV vector design, state-of-the-art manufacturing, and validated disease targets. Their vision was to generate strong promoters for cell type-specific expression in photoreceptors and not in retinal pigment epithelium. They wanted strong expression equivalent to the CAG promoter, which is a very strong constitutive expression, low CPG, and compact size, less than 1 kb. And so they needed a synthetic biology team that could design these promoters, develop the model cell lines to model photoreceptors and retinal pigment epithelials, and do high throughput screening and assay development. So again, the team of scientists that are now at GeneFab performed custom project onboarding, where they onboarded multiple model cell types and generated omics data in those cell types to be able to identify the transcription factors and open chromatin regions needed to design those libraries of promoters, and also developed custom transfection protocols to achieve high efficiency, high viability screening for library-based screening. And here we're showing the results of the assay, where on the left-hand side, you see the massively parallel screening, where 50 hits were identified from greater than 10,000 synthetic promoters that were highly specific for the on-target photoreceptors over the retinal pigment epithelium with 2 off-target cell types. And so we see that really, really highly specific promoter selection. And those were fed into iterative design and screening on the right-hand side, where you can see the first generation promoters are in blue, and the x-axis is showing the strength relative to CAG, where that green dot shows CAG at 1. And the fold specificity over the off-target cells is on the y-axis. So those first generation promoters were weak and achieved about 10 to 100-fold specificity for the on-target cells. But over multiple generations of design, the team was able to identify promoters with 1,000-fold specificity for the on-target over the off-target cells and strength equivalent to CAG, enabling strong and specific expression of therapeutic payloads only in the cell types of interest. And so what I've shown you through all these stories is that GeneFab can be an excellent partner for your next R&D project. We have synthetic biology services that are mapped to critical milestones in candidate selection, including the design and optimization of payload expression, customized cell and vector engineering, and high throughput screening in relevant therapeutic models. And we've proven through successful completion of multiple projects for leading industry clients that we can be valuable partners in the design of your next generation therapeutic. So we're looking to partner with cell and gene therapy companies that are looking to build their next generation product. We can do many things, including boosting or controlling expression, improving function, engineering safety, and manufacturing your product. And we're accepting new collaborations on a rolling basis, so please contact us now. And I'd just like to show some of a small subset of the faces at GeneFab, including our CEO, Philip Lee, our head of BD, Duncan Liu, myself, principal scientist on the synthetic biology team, Francis Liu, the director of BD and synthetic biology lead, as well as Pei Yi, senior director of BD, as well as our contact information, which is available below. And look forward to taking questions. Thank you.
Thank you so much, Dr. Hung. Looks like we do have time for a couple questions. If you haven't already, you can type those questions into the Q&A box. It looks like our first question is going to be requesting some more information about what is the future direction of your work.
Sure. I think we have a lot of future directions. It's often driven by clients, but we also do our own internal synthetic biology development. And so we're looking to develop, for example, UTR engineering for mRNA-based specificity to kind of complement the promoters that we have for DNA specificity, looking to further build out our master regulator discovery platform, and also looking to kind of make our current services faster and better, and just continue to improve.
Great, thanks. It looks like our second question is going to be, what promoters do you use?
Yeah, so the promoters, as you might have seen, they come from these huge libraries, which are built out of synthetic pieces, which come from, transcription factor motifs, enhancer regions, pieces of native promoters, and it's very customized for each client and their cell type or cell state that they're looking to specifically express their payload in.
Okay, great. Thanks. It looks like that's going to be all the time we have for questions. A big thank you to our speaker, Dr. Michelle Hung, for sharing GeneFab insights, and to all of you for joining us. We will begin our next session here soon. So thank you for joining us, and see you soon.